Cells perceive and respond to information and events in their microenvironment. A signal, such as a photon or a pathogen or a hormone, is detected by a receptor, which passes the information to the interior of the cell, where it is processed, and the cell can then respond to that information. For example, a cell detects a hormone and responds to that signal by initiating proliferation. Errors in cell signaling lead to diseases such as cancer, autoimmunity, and diabetes. So our ultimate goal in modeling cell signaling is to develop treatments for disease. The cell enters the cell through a receptor, then it is passed along by chemical modifications that proteins make on other proteins. A key modification is phosphorylation, wherein proteins called kinases transfer high-energy phosphate groups to other proteins, thus activating them. Generally speaking, at a given time, a signaling protein is in an active or an inactive state. A signaling event is the activation or deactivation of a signaling protein by another. Receipt of a signal triggers a signaling cascade, a chain of signaling events that delivers the message to where it needs to go in the cell. For example, a cascade activates a transcription factor, which then enters the nucleus and activates transcription of a gene. We can represent cascades of signaling events as causal networks. For example, in this well-known MAPK cascade, RAF phosphorylates MEK, or rather it activates MEK, and MEK activates ERK. So we get a causal network of these edges RAF-MEK and an edge MEK-ERK. This signaling network is the information processing mechanism that maps the initial signal to the correct cell response. Biologists have identified signaling pathways, which are canonical pathways of signal flow through the signaling network that are conserved across tissues and organisms. You can find these in pathway databases such as KEGG. In contrast, in computational systems biology, you go beyond just the causal relationships and try to detail the kinetics of the underlying biochemical interactions. These computational models are not as general, but they give you a deeper understanding of the system you are trying to study. In single-cell proteomics, you can simultaneously quantify the abundance of active state signaling proteins within a cell, giving you a cell-level snapshot of signaling activity. In fluorescence flow cytometry, active state signaling proteins are labeled with fluorescence tags, and fluorescence emission spectra are quantified. The cutting edge is mass cytometry, where active state signaling proteins are labeled with heavy metal tags. And the spectra is quantified when the cells are incinerated in a mass spectrometer. A quick note, some of you might be familiar with single-cell transcriptomics. The biology is obviously different than with proteomics, but from a statistician's perspective, another key difference is that single-cell transcriptomics datasets tend to have smaller sample sizes for cost reasons, while a mass cytometry dataset typically has measurement of 100,000 to millions of cells. A Bayesian network is a statistical model that represents a set of random variables and their conditional dependencies via a directed acyclic graph. The causal network representation of signaling that I just mentioned is the same as a Bayesian network representation under the constraint that the edge direction reflects causality. Structure learning algorithms are algorithms that infer Bayesian network structure from data. By applying these algorithms to single-cell data, we can essentially infer the signaling network structure directly from data. The workflow is like this. You first select the signaling proteins you want to quantify. You apply a set of perturbations to the cell samples. You apply a stimuli that activates the signaling cascade. You quantify the signaling protein using single-cell measurement technology like mass cytometry. Finally, you apply the network inference algorithm to the data. Now, the perturbations I just mentioned are actually quite important. What they do is they inhibit the ability of certain proteins to activate others. You need this kind of information in your experiment in order to get the causal interpretation of the inferred network. Otherwise, the edges will just reflect statistical association. For example, this inhibitor, imatinib, is blocking phosphorylation of that substrate, preventing downstream signaling. When you give this information to the algorithm, it can figure out whether things that are statistically associated with BCR ABL are actually upstream or downstream of it. So you can show this approach works by measuring some pathway that you know and then reconstructing the data. But in practice, reconstructing the pathway directly from the data using network inference. But in practice, you're not interested in just rehashing what you already know. You're interested in discovery of novel edges reflecting signaling events specific to your experimental context. For example, if you can discover specifically how signaling has been rewired in the disease cell population that you're studying, then you can determine how to drug those disease cells. However, this approach has an experimental design problem. Structured inference requires complete quantification of proteins involved in the signaling response. If you miss key proteins, you get erroneous edges. So you can't just measure all of the signaling proteins because the number of proteins that can be simultaneously measured is limited. Currently, with mass cytometry, that's limited to about 50 proteins. Experimentalists have to make choices about which proteins to measure, and there's no systematic method for doing so. We address this problem by building a computational model of the signaling system. We do this using two different inputs. One is prior single-cell data experiments that you have already collected, you've already done and collected the data on the same signaling system. And the other are canonical edges about this signaling mechanism from a database such as KEGG. From these inputs, we fit parameters of the computational model, and then using that traditional top-down systems biology approach, we look for parts of this canonical edge-based signaling network where our model doesn't match the data very well. We suggest proteins in this part of the network for quantification because they provide the greatest opportunity for discovery of context-specific edges.